Right. So it's 2. So we'll get started. And we're going to pick up right where we left off on Monday. So just as a review, what we were doing Monday, we've been looking at enzymes more in depth, and we started to look at different mechanisms. And then we ended on metalloenzymes, that is the role of metals in enzymes. And we just briefly talked about, how do enzymes actually speed up reactions? And we're going to pick up right on that topic and look at this a little more in depth um, when talking about our energy diagram. Remember, the energy diagram is right here, where we're looking at reaction coordinates versus free energy. Now, like we were saying at the very end of Monday, the way that an enzyme gets a good amount of energy to do all the orientation and placement effects is by interactions between the substrate and the enzyme. What I mean by that is that the substrate has different types of chemical groups on it, like charges or nonpolar or polar regions. And the active site of the substrate will try and match that. So when the substrate and the active site come into contact, that's favorable interactions, and that allows the enzyme to do some movement, right? Now, when we think about a reaction, a very simple reaction, there are 3 different states that the substrate goes through. First, we have the substrate itself. Then we have the intermediate. Well, actually, this is the transition state. And then we have the product. It can be a little more complicated, as I was about to write. You can have an intermediate state as well. But we're just going to stick with the basics, right? So 3 different states of our substrate. Now, an enzyme, there's 2 ways to think about an enzyme historically. One, if the active site is complementary to the substrate, so there's like a perfect bind there. Or 2, if the enzyme binds the substrate okay, but binds the transition site really well, right? So those are 2 different possibilities that historically people have come up with to try and explain the behavior of enzymes. So let's look at this free energy diagram versus reaction coordinates and see what the problem is with the first situation where the enzyme binds to the substrate. For this reaction in this image, which I think is excellent, again, this pulling across, our reaction is we're going to take this metal bar and break it in half. And our enzyme, the active site, is surrounded by magnets. So here in our first situation, our active site is perfectly shaped to bind to our enzyme. What that means is if we look at our free energy diagram, the binding of the enzyme substrate complex is incredibly favorable. So that we have substrate, and when it binds to the enzyme, free energy goes down because it's a perfect fit, right? All of the magnets are interacting with the substrate, which means if we want to get to our transition state, which is like the metal being half broken, you actually need more energy than if the substrate was by itself. Because if the substrate was just by itself, you can imagine taking that bar and bending it to try and break it. But here, when it's surrounded by metals, it's going to take even more energy to try and bend it. So this type of binding is actually slower than normal. or rather I should say uncatalyzed. Because remember, to determine that the energy of an enzyme or the speed, we care about delta G double dagger. That is from substrate to the top of your transition state, that energy gap. The bigger that is, the slower your enzyme. If we bind perfectly, though, we're actually increasing delta G double dagger. So we have actually slowed down our reaction quite a bit. But let's look at the enzyme that binds better to the transition site. So here, our active site, it binds the substrate okay, but not great. But what it really wants to do is to bind the transition state. And that's why we have this image over here, where the dotted line is the uncatalyzed reaction. And so by binding the transition state very well, we are lowering the free energy of the transition state. We are making that transition state easier to happen. And by lowering the energy of the transition state, we speed up the reaction. So in general, that's how enzymes work. They bind okay to the substrate. Then the enzyme actually changes. It goes through what's called a conformational change, where the active site will kind of move and force the substrate to look like the transition state, because it really wants to bind the transition state. Since we're lowering the free energy of the transition state, our reaction is able to speed up very fast. And this is why when we're looking at drug targets, a lot of drugs, antibiotics, are actually what are called transition state analogs. A transition state analog is a molecule that looks like the transition state, so it'll go and bind your enzyme, but because it's slightly different, the enzyme won't be able to do chemistry. The transition state will stay there forever, and you have just killed the enzyme. So that's why it's a good antibiotic or a good drug if you're trying to shut down some enzyme. But yeah, any questions about that idea of how enzymes want to bind substrates?
I'm gonna watch this. Hey, baby, I took the fish out because I'm gonna, I want
So there are two questions here. I'm just going to skip those for now, in that they're review questions from Monday. So make sure you do you go over those. The questions are, the answer to those are right in the PowerPoint as well. But I'm just going to move on due to time's sake to make sure we don't fall too far behind here in the very end of the semester. So let me get the new PowerPoint up, the one for today. And today we're going to continue to look at common mechanisms, or mechanisms of well-studied enzymes, I should say. And the first mechanism we're really going to look at is the lysozyme mechanism. So what lysozyme is, it's a very common enzyme, and it's found like in your tears and your saliva. And what it does is that it will go and destroy the peptidoglycan layer of bacteria. Remember, we talked about the peptidoglycan layer back in the sugar section of this class, where it is basically just a giant sugar protein net that's on the outside of the bacteria. And what lysozyme does is that it will cut right here between the D and the E sugar. So it will bind. So all these, let me take a step back there. And the peptidoglycan layer is made out of repeating disaccharides. These are called NAG and NAM. They're just sugars. Like, you don't need to worry about their names. We talked about them back in the peptidoglycan chapter, but we can just think of NAG and NAM as two different types of sugars, right? And so it just repeats NAG, NAM, NAG, NAM, NAG, NAM. That's the peptidoglycan layer with proteins hanging off the side. What lysozyme does is that it binds 6 sugars at a time. So it'll go and bind to 6 sugars. And the way it binds, it will distort the 4th sugar, or the D sugar. It will force it into the half chair conformation. What you need to know about that is that this is, it causes a steric hindrance. You're making the D sugar feel uncomfortable. Energetically speaking, this is an unfavorable conformation. And by doing this, lysozyme makes it easier to cut between the D and the E sugar, because you are inducing this steric effect. You are inducing this twist in the sugar. Yeah. There we go. Sorry, I usually do that at the beginning, but I guess it didn't work. For some reason, oh. Yeah, so that's plugged into my headphones. That's why I couldn't hear anything. Thank you for letting me know. But yeah, we're gonna cut between the D and the E sugar because of the sterics we are introducing in the D sugar. And we're gonna begin today by looking at this mechanism. So this type of mechanism is going to be like acid catalysis. And here we're just seeing what's going to happen. Before we actually look at all the arrow movings, this slide is really just preparing you for, what type of reaction is going to happen. So the way that we're going to break this ring is that we're going to convert our acetal to a hemiacetal. So if you remember, the way that sugars are connected is through a glycosidic bond, which is being shown right here. So this R double dash or double apostrophe, this is the other sugar. So we're connecting through our glycosidic oxygen here. The idea behind this is that this oxygen is going to get protonated. Once it gets protonated, this oxygen is going to be cut off and more or less replaced with water. So you'll have a hemiacetal with an OH. And then your other sugar molecule will no longer be attached. And our transition state is an oxonium ion, where we will have either a positive carbon, a positive oxygen or a positive carbon when we do this. Either one of these is not favorable, right? You don't want a positive oxygen. You really don't want a positive oxygen. And you don't want a positive carbon either. Neither of these molecules like to be positive. So the idea is, from what we talked about at the very beginning of class, and then a little bit on the last slide, to cut this peptidoglycan layer, we're going to look for the enzyme to try and one, make this oxonium ion, and to stabilize it so it's not so unfavorable. So let's take a look at this. So here is our mechanism. The players, the amino acids that we really care about here are GLU35 and aspartic acid 52. So quick question right off the top. Does anything look weird about GLU35 or ASP52? Based on how I taught you the amino acids, which one of these is drawn a little weird? Which one of these looks not normal? Glue is protonated. Yeah, that's really weird. That is very uncommon, right? Glutamic acid has a pKa of like, what, 4.25? So it really does not want to be protonated. But lysozyme, and I have this on another slide, lysozyme, the amino acids around GLU35 have changed the pKa of GLU35 so that it will want to be protonated. It's a really cool phenomenon that happens in enzymes all the time. You can change the pKa's of amino acids based on what other amino acids you have around it. But yeah, that's just the weird thing. GLU35 is protonated to begin with. All right, step one, we bind our 6 sugars. So not shown here is A, B, C, and F. So they're binding, but the only 2 sugars that are really going to do chemistry is D and E. So like in our previous slide, this glue is going to act like an acid, and it's going to donate our hydrogen to our glycosidic bond. So that's the first step, and just like on the previous slide. And we are making our oxonium transition state. So here we have our positive carbon now, which is unfavorable. So to stabilize this, we have As52. And what As52 is going to do is covalent catalysis. It's going to go and bind to our D sugar so that it's no longer a carbocation. And instead, it's connected to our lysozyme, right? So that is how we're really speeding this up. That these 2 steps kind of happen in concert. One, we give a proton to our glycosidic bond to break it. And then at the same time, we're going to be binding this oxonium ion. And the whole time too, As52, it's not really shown here, but that negative oxygen is going to be near this carbon the whole time. So that carbon doesn't mind going positive because it has a negative amino acid right next to it. So that is how we're trying to stabilize the transition state. Once we have our covalent catalysis, we have to reverse the reaction, right? We have to get glue back to its protonated form, and we have to remove this sugar from As, like we talked about on Monday. When you do a reaction, you kind of just have to do the reverse of it. So what's going on here is now base catalysis, where GLU35 is going to be acting like a base. And it's going to take this hydrogen from water. Water at the same time will attack our former carbocation, or the carbon where we had our glycosidic bond, or the anomeric carbon. Each one of those names would fit. And that OH is going to attack. Once that OH attacks, then asparagine can break its covalent bond to the sugar. So now our other sugar is free to leave lysozyme. As52 is back. to being unprotonated, glue 35 is back to being protonated. And then you can start again. And that is how lysozyme creates the oxonium ion, stabilizes the oxonium ion to speed up the reaction of breaking sugar glycosidic bonds. So questions about that? Right. So predict the effect of lysozyme's activity on mutating glue 35 to ASP and ASP 52 to glue. So this is a good one of those like critical thinking problems where the answer is not obvious. And although I've kind of taught you what the answer is, you kind of have to go digging back in your memory to try and rationalize this. So I think this is better to look at if I have this up. So the question is, what happens if instead of glue, this was ASP, and instead of ASP, this was glue? So to really tackle these types of questions, I find it very useful to like draw pictures. So if this was ASP, we would lose one CH2, and then our carbon C double bond O would be on this carbon. And if this was glue, well, we just move that over. And so the question is, what would be the problem with that? And the problem most likely is that the enzyme probably would not work. One, who knows if this ASP would be protonated, because aspartic acid has a lower pK than glutamic acid does to begin with. So it could be, even though that the environment wants a glue to be protonated at position 35, maybe it would not work for an ASP. And the other problem would be sterics, or distance rather. This activity, this enzyme, this has evolved over millions and millions of years, right? So it's really, really good at what it does. However, if you remove a carbon length, now your C double bond O will be over here, and it might be too far to interact with that oxygen. And since you're increasing ASP, maybe that oxygen is too close to the carbon, that the carbon would not want to go near it at all due to steric hindrance. So that's another way that you could kill the enzyme. And so just in recap, the enzyme probably would not work if you just switch around the ASP and the glue. One, ASP probably won't be protonated, and it needs to be. Two, the distances would probably not work out anymore, and you would probably incorporate steric clashes there. But yeah, that is I really like questions like that just because it makes you think, and it's just not pure memorization. Question 2, it's more of a review. We kind of hammered that home already. So yeah, just when you're going back to study for the test, make sure you can answer question 2. And it has to do with, do you know the mechanism of lysozyme? That's really what this question is designed to make you think about. And then more lysozyme questions. And hopefully by now, since I've asked 3 questions on lysozyme, you can probably be pretty certain I'm going to ask you questions on the mechanisms of lysozyme. So here we have residues D101 and R114. They're required for catalysis, but they are further away from glue 35 and AS 52. So I dropped, it's not glue 3, it's like glue 35. So if you replace alanine for D101 or R114, the enzyme looks the same. So the enzyme tertiary structure does not change. So it looks the same, but the activity goes way down. So explain this. And this is a phenomenon we see in enzymes all the time, right? So when you study or look at enzymes, usually people are just focused on the active site. They're just focused on the amino acids directly surrounding the substrate. But that's not the whole picture. What could be happening is through, ionic interactions or hydrophobic interactions, there are amino acids that are far away from the active site that still influence the chemistry. And that's what's going on here. Even though ASP101 or arginine 114 are not close, they still can interact with the active site through, here would be electrostatics, most likely, or hydrogen bonds. Because they're charged or because they can form hydrogen bonds, they are interacting with the active site through long range interactions. Change that to an alanine, you kill those interactions, right? The enzyme itself, the shape doesn't change, but you now are losing an interaction in the active site. And that would slow down the reaction. You are losing the ability to stabilize that transition state for some reason. We might not know what the reason is. It's not all that obvious, but that's what's going on here. So questions about that explanation or anything up to this point? All right. The next type of enzyme we're going to look at is called trypsin. And what trypsin is, it's a molecule that's part of a class of enzymes called serine proteases, right? So what a serine protease is, it's a enzyme that chops up proteins. And the most important, well, the amino acid that's like directly involved with this is serine. But there are 3 critical amino acids in every serine protease, and they're called the catalytic triad. That would be aspartic acid, histidine, and serine. So these 3 amino acids, and they're called the catalytic triad because in all the different serine proteases that you discover, that or have been discovered, we always see these 3 amino acids working together, right? So they're very important for the function of this enzyme. Serine proteases have a lot of different roles in the body and inside cells, as I have here. And 3 of these have been studied very well, chymotrypsin especially, trypsin, and elastase. And we're going to actually look at how these serine proteases work as another deep dive into a specific enzyme. So first, we're going to look away from these catalytic triad, and we're going to look at what's called the specificity pocket. Now, for each serine protease will chop a different amino acid. And what I mean by that is when we are doing a reaction with serine protease, their reaction is to chop up a peptide bond. But they will only chop after certain amino acids. And what amino acids that they'll chop depends on their specificity pocket, right? So let's look at the 3 types of well-studied serine proteases again. So we have chymotrypsin, trypsin, and elastase. Now, chymotrypsin cuts after aromatic side chains. And if we look at the specificity pocket, and this is where our substrate will bind, you can see that we just have glycines, so very small, and serine, again, very small. So more or less a hydrophobic pocket that does not have big side chains, has very small side chains. So a big bulky group like phenylalanine can go right in there, no problem. And then we can cut right after our bulky aromatic side chains. Trypsin, on the other hand, cuts after positively charged amino acids. And like chymotrypsin, we have 2 glycines in the same spot. But the big difference here is that instead of serine at position 189, we have the negatively charged aspartic acid at position 189, which is causing this favorable charge interaction to happen, where we have our positive amino acid interacting with our negatively charged amino acid. Once we have bound in the specificity pocket, we cut after the positive amino acid. One thing to note, and it's interesting, and it goes back to the previous question I just asked. If you look at the specificity pockets between chymotrypsin and trypsin, the only thing different is at position 189, serine to aspartic acid. However, if I take serine and chymotrypsin and I mutate it to aspartic acid, you actually don't make trypsin, right? You just create a protease that's non-specific and just really bad at its job. That is, once I mutate this serine to aspartic acid, I'll just cut random stuff, but I won't do it very fast, which is really odd. Again, if we're only focusing on moving arrows, if we're only focusing on that part of the enzyme, you would not expect that. So this is just another reminder that the amino acids in enzymes, even though they might not look important because they're not directly involved in the mechanism, they could still be influencing the active site through long distance interactions. Our last specificity pocket is elastase. What elastase cuts after are alanine, glycine, and valine. So small hydrophobic amino acids, and in the pocket we have small hydrophobic amino acids. So again, helping that bonding there. So that's a review on serine proteases. They cut proteins, and the 3 types of serine proteases that are major. Now, I do have a mechanism for the catalytic triad. However, this test is big on mechanisms already. So for the sake of the serine proteases, we can just skip the mechanism. Like, I'm not going to ask you questions on it for that. Lysozyme, that's free game. The mechanisms we talked about on Monday, the concerted acid-base mechanism, that's free game. I'm going to save you some trouble here at the very end of our spring COVID semester and just say, yeah, we can skip this. If you'd like to actually know how the serine protease works, it's here for you. It's in your book, but we're going to skip it just for test purposes, the actual arrow moving. So I won't ask you questions on that. What I do want to go over, though, is transition state stabilization in serine proteases. So again, this whole idea of why enzymes work. So our intermediate here is we're breaking a peptide bond. And to do that, we're going to create a negative oxygen, right? So this is our transition state. We have, we see this transition state a couple times in the mechanism. But the big thing here is we're gonna take a normal peptide bond, which looks like this. If you remember that. And we are basically, it actually goes NH here. And we're basically gonna make this carbonyl carbon go negative. So this is the only part we're really focusing on. We're gonna make that go negative by, we'll have another oxygen connected to it. This is what we're trying to stabilize. This is our transition state right here. And the way we do that is through what is called the oxyanion hole. So remember, the three amino acids of the catalytic triad are serine, histidine, and aspartic acid. They're going to be doing the main chemistry. But to stabilize the transition state, there's an area in our enzyme next to serine 195 and glycine 193. So both of these amino acids are showing their N-termini, the NH bond. They're both pointing to empty space in the enzyme. And what happens is when serine 195 bonds to our backbone and makes a covalent bond between the carbon and the oxygen, that carbonyl oxygen becomes negative, but it is stabilized by hydrogen bonds from serine 195 and 193. So that is the purpose of the oxyanion hole in serine proteases, is to stabilize a transition state, and it is created by the N-terminal, or sorry, the amide backbone groups of serine 195 and glycine 193. So that is how we're going to speed up the cutting of a peptide bond by stabilizing the transition state. Right. Questions about that idea? Right. Now, trypsin and all the serine proteases, their job is to cut up proteins. Your cells are full of proteins. And so it would be very bad if these enzymes, when they were created from the ribosome, were automatically turned on. Because what they would do is they would go around your cells and just kill all your proteins. We don't really want that. So any enzyme whose job is to chop up stuff, in this case, chop up proteins, they are usually made as bigger versions than their final products. And these are called zymogens or proenzymes. And what a zymogen is, it's a version of a proteolytic enzyme that is inactive, right? So the zymogen of trypsin is called trypsinogen. And it's just the original version of the enzyme that is not active. People suffering from acute pancreatitis, as written there, that happens when you make these enzymes, but they're already turned on before they're supposed to be, and so they just kind of eat away at the cells. So that's really bad. And the way that we turn these enzymes on is actually called an, well, we need an extrapeptide. One is called enteropeptidase. And what enteropeptidase does is that it will cut between lysine 15 and 16. And then these amino acids will fly off. So lysine to the N-termini will go off. And it's actually what's shown here is a hexapeptide, where so this is lysine 15, 4 aspartic acids, valine, the N-terminite. So before we even talk about this, residues 1 through 9 were already chopped off. And so we're left with 10 through 15 at the N-terminus. Enteropeptidase cuts between 15 and 16. Or trypsin can actually do this, which is called autocatalytic response. So if there's an active trypsin around, what that trypsin will do is that it will go and find trypsinogen, which is the precursor to trypsin, make that cut and create more trypsin, right? It's kind of like a chain reaction at that part. If you have one trypsin activated, it will activate other trypsins, which will go and activate other trypsins. So it's very important that we control this process. And so like enteropeptidase, that would only be present where we want trypsin to be activated. If we don't want trypsin to be activated, we don't have enteropeptidase and we can't start this process off. But yeah, that's a zymogen, and that's a very common technique to control our proteolytic enzymes. Now, that's basically all I have for today for our slides. So what you want to focus on for the test that's coming up next week, make sure any mechanisms that we talked about, that you could repeat those back to me and you know what are the point of the amino acids and being in those positions. I'm going to ask you questions about that. Make sure you know how these enzymes stabilize the transition state, and you know why that's important based on the very first slide we talked about today. And also, what is the zymogen? Why isn't trypsin always active? So make sure you know about that. Otherwise, we can end a little early. And on Friday, as I noticed on the folders, we aren't completely synced up to the folders anymore. So on Friday, we should be able to sync right back up and be where we need to be at. Otherwise, yeah, I have nothing left for you. So if you have any questions, please don't hesitate to email me. Happy to get through those. I'll put a homework up so you can review what we talked about. Otherwise, thanks for stopping by. Thanks for watching YouTube. And hopefully I'll see you on Friday. Have a good day.